From the JAMA Network, this is JAMA Psychiatry Author Interviews. Conversations with authors exploring the latest clinical research, reviews, and opinions featured in JAMA Psychiatry.
Hello, and welcome to our listeners around the world. You're listening to the JAMA Psychiatry Author Interview podcast. I'm your host and web editor, Dr. John Torous. Today, we're thrilled to be discussing a new paper just out in JAMA Psychiatry, the 25-year evolution of lithium as a disease-modifying agent in dementia, from BDNF, BCL2, GSK3-beta to brain repletion, a narrative review. We're about to be joined by the corresponding author of the paper, Dr. Manji, from the Department of Psychiatry at Oxford University in Oxford, UK, and the Department of Psychiatry at Yale University in New Haven, Connecticut, USA. Lithium has been a cornerstone of psychiatric treatment for 75 years, but for most of the time it was primarily thought of as a mood stabilizer. But over the last quarter of a century, that picture has changed. A growing body of evidence suggests that lithium does something far more fundamental. It may also strengthen cellular machinery that keeps neurons alive, healthy, and connected, and that has raised brand new possibilities. Could lithium also slow cognitive decline in illnesses like and related to Alzheimer's disease? A new narrative review in JAMA Psychiatry traces this 25-year evolution of this idea and its new clinical implications. So to learn more, we're about to talk to the corresponding author of the paper. So thank you so much for joining us, Dr. Manji. To jump right in, as we said, lithium's been around for 75 years. How has the field moved from thinking of lithium purely as a mood stabilizer to something new that maybe has neuroprotective properties?
Thank you very much, John, for the opportunity to talk about this very interesting work. As you correctly pointed out, lithium has been around a long time, and for the longest time it was viewed as a mood stabilizer. A lot of research then started to show that lithium affected pathways inside the neurons. And so one of the questions I had was, what does that lead to? As you and the audience probably knows, there are now new technologies to figure out what genes are turned on or off in disease states or with treatments. And we used to have to look at our favorite gene, this serotonin gene, this dopamine gene, etc. But now we have ability to look at all the genes. So it's a method called an unbiased view. So you're not looking for a specific gene, you're asking the question, what genes are turned on or off with treatment? A lot of those studies were done, and somewhat to my surprise at the time, one of the genes that was turned on was BCL2. I'll come back in a moment to what it is. When you see genes are turned on or off, you need to validate it at a protein level as well. And so that was done as well. And the data showed that lithium robustly upregulated BCL2. And importantly, this was occurring at sort of what we would consider therapeutic concentrations of lithium and over a several week time frame. Now, one of the reasons that it was surprising that BCL2 was turned on is that BCL2 is widely regarded as the major neuroprotective protein. Interestingly, we have a lot of BCL2 in the developing brain, then the levels go down. So the idea that you could turn up BCL2 in the adult brain was very exciting. But then that begged the question, could this be relevant for bipolar disorder? Because that's not Alzheimer's disease where a lot of nerve cells die. But a lot of data was showing that when you do MRI scans of patients with bipolar disorder, you see what looks like shrinkage or atrophy. And so this suggested that BCL2, if it was getting turned on, could actually be very relevant to provide neuroprotection in patients with bipolar disorder.
And your paper identifies these three molecular targets. We talked a little bit about BCL2. There's also BDNF, and there's GSK3-beta. For listeners who aren't as familiar, let's do a quick mini review of kind of why these three targets, again, BDNF, BCL2, and GSK3-beta, are important for brain health overall.
Very good question. So lithium was also found to turn on BDNF, which stands for brain-derived neurotrophic factor, And it's a very important molecule in what I call neuronal resilience. So it helps neurons sort of survive against different kinds of stresses and insults. It's involved in what people consider neuroplasticity and synaptic integrity. Just coming back to BCL2 for a moment, it's thought to exert most of its effect at the level of the mitochondria and inhibit these things called cell death cascades. So that serves a major neuroprotective role. And then GSK3 beta is one of these enzymes that, when it's overactive, it sort of promotes cell death. And with respect to Alzheimer's, it phosphorylates this molecule called tau, and tau hyperphosphorylation is thought to be bad in Alzheimer's, and also increases the level of amyloid deposition. So through these pathways together, we provide a lot of neuroprotection to the brain.
Yeah. And this is amazing because, again, in medical school, we all learn lithium, bipolar mania, suicide prevention, but this is a whole different story we're talking about, neuroprotection. To dive into some of these benefits that you're seeing, can you talk about lithium's effect on mitochondrial function, oxidative stress, and why is, again, helping mitochondrial preservation so important for neuroprotection?
Another very important question. So, as I'm sure most of your audience is aware, neurons, nerve cells, are very metabolically active. So, some studies suggest that about 20% of our energy utilization is occurring through the brain. And as a result of that, you have to constantly replenish energy. And so the neurons in particular are more susceptible to sort of an energy drain, for lack of a better word. And mitochondria, they're basically the energy powerhouses of the cell. So what lithium does by upregulating BCL2, it optimizes ATP production, which is the energy molecule. It prevents the release of molecules like cytochrome C, which are involved in cell death. And as a result of this effect on increasing mitochondrial energetics, it's almost like a final common pathway. So people, until lithium was shown to bring about this effect, there was no pharmacological way to increase BCL2. So scientists have genetically engineered rodents to have more BCL2 in the brain. And when they do that, they see protection against a lot of insults, including oxidative stress, including ischemia, including Parkinson's or Alzheimer's toxins, and glutamate excitotoxicity. So the upregulation of BCL2 has a major effect on mitochondria, and as a result of that, it protects against a variety of insults.
Which is a pretty good function to have more of your energy powerhouse and less of the destruction properties now. And let's switch a little bit to the neuroimaging evidence from humans. What did early imaging studies show us, and how does that kind of help build this case of the neuroprotection properties of lithium?
So again, a very important question. And I think, as we know, sometimes you can see all kinds of effects in rodents, but do they actually happen in humans? So that was one of the things I wanted to test with, when lithium upregulated BCL2. So using magnetic resonance spectroscopy, that allows you to measure certain chemicals in the brain. And a very important chemical you can measure is called N-acetyl aspartate, or NAA. It's thought to be a marker of neuronal viability. So not whether neurons are just alive or dead, but how healthy are they? And certainly if you look in conditions where neurons are sick, you see the NAA levels going down. So what we did was did a longitudinal study in bipolar patients. So these are patients with bipolar disorder. They happen to be in the depressed phase because it's easier to do MRI scans when someone is not manic. We did the baseline studies to say, okay, of all medications at baseline, what are your NAA levels? And then we put them on lithium for 4 weeks and then remeasured NAA levels. And we found that the NAA levels were consistently increased,
Oh, wow.
suggesting that similar to what we saw in rodents, you're basically increasing neuronal viability. So those were very interesting findings. But then we wanted to look at structural changes in the brain. As I mentioned a moment ago, many patients with bipolar disorder or major depressive disorder have shown some reduction in volume. What was a very interesting observation was that in the hippocampus, people had seen shrinkage or atrophy in major depressive disorder, but not as consistently in bipolar disorder. And I think we have the explanation. So, what we did was once again take bipolar depressed patients with no medication, did the MRI scans to quantitate the amount of gray matter in parts of the brain, put them on lithium for 4 to 6 weeks, and measured it again. We also did a healthy volunteer study at the same time, and I'll explain why. And in the healthy volunteers, again, treated them identically. So the findings were that in bipolar disorder patients, in the regions of the brain that had been reported to have atrophy, we saw a reversal of the atrophy. So we saw an increase in the gray matter volumes. The healthy volunteer study was also important because it showed us that lithium wasn't acting just like a non-specific brain fertilizer. It was actually reversing atrophy due to illness. So in the healthy volunteers, we didn't see this effect. Coming back to what I said about the bipolar disorder brain, we spoke to a number of investigators who had done a lot of MRI studies in bipolar disorder patients, and we asked them to reanalyze their data based on whether people were on lithium or not on lithium. And the answers were very consistently the same. If you were on lithium, you did not show this atrophy, whereas if you weren't on lithium, you showed the atrophy. And that seemed to explain these sort of inconsistent results in bipolar disorder. It was actually due to treatment. If you were on lithium, you had the preservation of the brain volume. If you were not on lithium, you had the atrophy.
This is remarkable because we've gone from, again, molecular evidence, spectroscopy, we've gone to MRI. Let's now talk about the kind of epidemiological level evidence at a more population level, and how does this kind of inform about lithium and dementia risk and the neuroprotective properties?
Again, a very important question, and especially in European countries, they've done a lot of epidemiological, naturalistic, and registry based studies. So, several groups reported that individuals on long term treatment had lower than expected rates of dementia compared either to the general population or to bipolar disorder patients treated with other agents, not lithium. And then one of these studies, it was a large Danish registry, they found that cumulative lithium exposure was associated with significantly lower rates of dementia. And so this, what I mentioned, cumulative lithium exposure, the longer you were on lithium, the greater the reduction in dementia risk. And these findings have been replicated in other Scandinavian and UK data sets. One other intriguing finding, but you have to be careful about interpretation, was that ecological studies comparing lithium concentrations in drinking water found lower dementia prevalence in areas with more lithium in the drinking water compared to not, suggesting that low levels of lithium might also be exerting some benefits. But obviously, you need to be careful about, sort of amount of lithium in drinking water and dementia.
Now, you've put together all these pieces again from molecular evidence, spectroscopy, imaging, epidemiology. In randomized clinical trial evidence in mild cognitive impairment, MCI, and Alzheimer's, been a little bit more mixed on what is the impact of lithium. What do you think is driving these mixed results? And what do more recent low-dose trials suggest in being low dose of lithium?
Yeah, so I think that's again an important question. I think most people, most of your audience knows that dementia is this long process where, the pathology starts maybe 15 years before you get dementia. And then, you get the progressive increase in pathology and the dementia. And a lot of evidence today suggests that if you try and treat at the dementia stage, it's almost too late. The neurons are dead. And so I think some of the negative results were because they looked at people with full blown dementia. Some looked at mild cognitive impairment, but others looked at people with full blown dementia. And the other important thing is that these effects of lithium very likely take a long time to work. So you start to upregulate BCL2, for example, within 4 weeks. But then the upregulation of BCL2 has to sort of counter the Alzheimer's pathology. So some of the studies had also looked at a much shorter time duration. And I think it's important to both intervene early in the disease stage and to look at disease progression over months rather than years if you really hope to see a beneficial effect. This year, there was a 2-year pilot study, again in dementia, which did not show, meeting the primary endpoint, but it significantly slowed down the decline of verbal memory, again suggesting that if you design the study well, intervene early, and follow people long enough, you will hopefully still see an effect. What you mentioned about lower dose is very important because, as you and your audience knows, lithium has what we call a narrow therapeutic index. So it's got to be at just this right amount. And if you go higher than that, a lot of people have side effects. But even at regular bipolar disorder, many people have side effects and don't like it. So in my lab, I did a lot of studies with a lower dose, so doses that gave you blood levels of about zero point 0.3 millimolar, and the bipolar doses are thought to be zero point 0.6 to 1.0. And we found that even at these low doses, we found these neuroprotective effects in rodents. And I think that's very important because that suggests that while you might need a higher dose for anti-manic or antidepressant effects, to provide this neuroprotective effect, you can get away with a very low dose. And if you're using a very low dose, very likely the risks for renal or thyroid dysfunction, or in the elderly, sort of they might be a little bit more susceptible to even cognitive effects. Our thinking is that you're getting to this very low dose lithium, it's going to be very well tolerated and you're not going to get side effects. So you can do the trials in elderly people and hopefully show that they have the benefit and it's got very good tolerability.
That could be a paradigm shift in how we treat these illnesses. And in the paper, again, and here we've talked a little about Alzheimer's and mild cognitive impairment, but you also discussed lithium's potential across other dementias, Lewy body, frontotemporal, vascular. Is it kind of the same mechanism working, or are there differences when we kind of talk about these other dementias?
So again, very good question, and I think it's both. So in some ways, because lithium is turning on some fundamental neuroprotective pathways, it has the possibility by this general neuroprotection, but to provide protection against a variety of dementias. But there are also some specific mechanisms. So say if you start with diffuse Lewy body dementia, lithium enhances something called autophagy. And autophagy is a process that the brain and other cells sometimes use to chew up and get rid of some bad proteins. So in diffuse Lewy body disease, one of the proteins that builds up is something called alpha-synuclein. And lithium, by enhancing autophagy, has the capability of sort of helping chew up the alpha-synuclein. In frontotemporal dementia, one of the problems is thought to be that GSK3 beta phosphorylates tau, and abnormally phosphorylated tau leads to the formation of something called neurofibrillary tangles. Lithium is both directly and indirectly inhibiting GSK3, which should hopefully help here. And just the last example to mention is in vascular dementia, the mitochondrial protection is very important. And then lithium also seems to turn on some anti-inflammatory cascades, which would hopefully also help against ischemic injury.
No, it's quite remarkable, all the potential. And maybe my final question would be, looking ahead, what's your take on, again, the evolution of lithium and maybe just where we're going? Clearly, you've outlined this amazing research you've done, the potential. What do we all look out for as the audience listening and hoping to benefit from this fantastic work that you and your team have done?
Thank you. So I think the data is looking very, very promising. But even this last year, there was this intriguing finding from Harvard. So this group at Harvard basically took post-mortem brain from healthy controls, from patients with mild cognitive impairment and patients with Alzheimer's disease. And they looked for the levels of multiple metals, copper, zinc, magnesium, lithium, etc. And one of the ones that was found to be very low in the Alzheimer's disease brains was actually lithium. And they had blood levels of lithium, and they saw that those were normal. So for some reason, the brain levels were low. Then they asked the question, is it all the brain? And so they did a very sophisticated study. As your audience knows, in Alzheimer's disease, you start to build up these things called amyloid plaques. And amyloid plaques are very negatively charged. And these investigators were able to show that the high negative charge sort of ties up lithium. So you have much lower levels in the rest of the brain. And so that would suggest that, sort of a lithium deficiency in the brain could be promoting the progression of dementia. What they also did, which I think is a very intriguing finding, is they were able to show that traditionally when I went to medical school, and probably you and your audience, we thought lithium is lithium, whether it's lithium chloride, lithium carbonate, etc. But there's another form of lithium called lithium orotate. And the reason it might be different is that it actually binds more tightly to lithium. So there's some data to suggest that lithium orotate is transported into the brain at higher levels than traditional lithium. And the lithium orotate doesn't bind to amyloid as much. So I think there's two advantages there. One, not binding to amyloid as much would give you more lithium left. And if you can get a higher brain to periphery level of lithium, you could even go with a lower dose, because as I mentioned, the studies I did were using a blood level of 0.3. If you could get away with it an even lower level and get the same amount in the brain, that would be remarkable. Now, the cautionary note I'd want to mention is I think this was a fantastic study that was done, but it is a study, so we need to look for independent replication. But I think if I was to summarize all the data, I think you're going to see, and in fact, I'm hoping to do that myself, is studies of low-dose lithium and perhaps specifically lithium orotate in looking at whether you can treat individuals with mild cognitive impairment and slow down the progression to Alzheimer's disease. The way we're going to do these studies is first we're going to take rodents and treat them with either lithium carbonate or chloride or lithium orotate and measure the levels of lithium in the brain. Is it true that you get more lithium orotate in the brain? And in rodents, you can use something called a 16 Tesla magnet, which is a very powerful magnet. Then we will do a human study in a similar way, lithium carbonate or orotate, to see what blood level of lithium do you need to get the right amount in the brain. And then with the data, we will do a study in mild cognitive impairment, where these are people who have these, mild cognitive impairment. And there's now a blood test which is very accurate, which could tell you whether you're on your way to dementia or not. So people with mild cognitive impairment who have this biomarker, then they would be randomized to lithium orotate or placebo and followed over 2 years to see have we in fact slowed down the progression of dementia. And if it works, you would potentially have a treatment which is dirt cheap, available everywhere in the world. And if you're using this very low dose and you don't have the potential side effects, you might even be able to get away with very infrequent blood testing because you don't need it. So it could be really useful for the whole population.
Oh, that's a fantastic study. Hopefully we'll get to cover it in JAMA Psychiatry, and I'll make a corny psychiatry joke of it being dirt cheap since you can mine lithium from the dirt. So maybe on that corny joke we'll end. And thank you so much, though, for sharing this remarkable body of work. And of course, there's so much more in the paper to read. We tried to cover the highlights, but this is a fantastic paper. And again, I'm John Torous. I've been speaking of Dr. Manji about the 25-year evolution of lithium as a disease-modifying agent in dementia, from BDNF, BCL2, GSK3 beta, to brain repletion and narrative review. To learn more about this research, you can find a link to the article in this episode's description. To follow this and other JAMA Network podcasts, please visit us online at jamanetworkaudio.com or search for JAMA Network wherever you get your podcasts. This episode was produced by Daniel Morrow at JAMA Network. Thank you for listening.
This content is protected by copyright by the American Medical Association with all rights reserved, including those for text and data mining, AI training, and similar technologies.